Hello, my name is Dylan Poach, and I'm working with Tyler Sodia under Dr. Andrew Bonham on researching electrochemical DNA biosensors for detecting non-tuberculosis mycobacteria. So, what are mycobacteria? Mycobacteria are a widespread infectious bacterial agent. There are many different types of mycobacteria. One of the ones that you might be familiar with are Mycobacterium tuberculosis, which is a cause of TB disease. Additionally, there's Mycobacterium leprae, which is a primary cause of leprosy, and a lesser known type, which is what we're interested in, non-tuberculosis mycobacterium, or NTM. So non-tuberculosis mycobacteria have been identified as many different species. There's over 140 different species that have been identified so far. Each and every year, roughly $1.7 billion is spent on mitigating NTM disease-related costs. Additionally, we're seeing a severe clinical increase in NTM lung disease cases, with nearly 100,000 cases in the United States alone. Currently, the gold standard remains culturing or sputum and lung lavages. Unfortunately, these techniques take about 6 to 8 weeks to render a good diagnosis, which can be a very long time for someone who's afflicted with this disease. As you can see down here, there's a CT scan of a patient's lung who is actually infected with NTM-LD. So to detect NTM, it's a very long process that can take 6 to 8 weeks. So many people are pursuing alternative methods of diagnostics, in which include the biosensor. So biosensors are devices that detect molecular targets using a biological-based sensing mechanism. Typically, a biosensor is made up of 3 different components. One, you have an analyte. This is a thing that you're trying to detect. It could be a protein, it could be a toxin, it could be many different things. Then you have the bioreceptor. This bioreceptor is going to sense the analyte and basically emit some type of signal. There's many different types. It could be optical, acoustic, immunoenzymatic, electrochemical, which is what we use, which is going to go to this transducer. And once this signal gets picked up by the transducer, it emits some type of measurable output that's put into a detector and gives us data where we can analyze if that analyte was actually in the sample or not. So what we use is electrochemical DNA, or eDNA biosensors. For eDNA biosensors, the bioreceptor is an aptamer sequence, which we'll talk about in a moment. It's basically a single or double-stranded DNA nucleotide sequence. And then for the transducing element, we use voltammetric interrogation. There's many different types that can be used. What we personally use is square wave voltammetry. A lot of people use cyclic voltammetry, but our lab has developed many different eDNA sensors. So far, we've developed sensors to detect ricin toxins, botulism toxins, and even celiac disease in buffered solutions. Here is a schematic of a typical eDNA biosensor. As you can see, there's a nucleotide aptamer sequence with some type of redox moiety. What we use is methylene blue. And then this aptamer is going to bind to some type of analyte and cause a conformational change that is going to move this redox moiety closer to the gold surface electrode. And that's going to give us an output of some type of electrochemical current that we can then go and analyze. And here's just a fun photo of the actual electrode that we use in our lab. So I've used the term aptamer quite a bit. What is an aptamer? So an aptamer comes from aptus, from Latin, meaning to fit, and the Greek meros, meaning part. So basically an aptamer is a nucleotide sequence that's going to fit and bind to a certain molecule, a certain target. An aptamer is a specific sequence and it can bind to anything from proteins to carbohydrates, toxins, etc. We generate these aptamers using a process called SELECT. SELECT stands for Systematic Evolution of Ligands by Exponential Enrichment. I know it's a mouthful, but basically, SELECT is a process where we take a target molecule, we put it into a library of billions of nucleotides, and the nucleotide sequence that binds to that molecule with the greatest affinity is going to be our aptamer sequence. Over here is a schematic of an aptamer sequence. It can take on these three-dimensional formations and become a functional aptamer. And upon binding to a target, it's going to change that conformation to allow efficacious binding. So we use two different types of molecular targets: manocylated liporhabdomanins, or MANLAMs for short, and glycopeptidolipids, or GPLs. MANLAMs can be represented by this chain of orange circles right here that goes down to the plasma membrane, and then GPLs can be represented here by this blue rectangular object, and that just kind of goes down to the outer membrane. It doesn't permeate through the plasma membrane. So the first target that we started with was actually MANLAM. MANLAMs are a glycolipid that are found in many pathogenic species of Mycobacteria. As you can see, it's heavily glycosylated and consists of this mannan backbone and a rhabdonin core down here. This structure is just a typical liporhabdomannan, but what makes our MANLAM, the actual mannose caps, is that the mannose caps are found on this rhabdonin core down here. There's many different types of lambs. You can have pi-lam, you can have aralam, but those are going to have like phosphate inositols and no caps respectively. So there's many different types, but we chose to investigate MANLAM as it's very prevalent in the key pathogenic species. The second molecular target, GPLs, is possibly an even better target as it is NTM specific. This means you're not going to find it in just tuberculosis or Leprae. You're going to find it only in non-tuberculosis Mycobacteria. Unfortunately, I do have to point out this is a very amphiphilic molecule, meaning it has both hydrophobic and hydrophilic ends on the molecule itself. This is very difficult when trying to purify this GPL structure using TLC plates, and it makes it very difficult to find the right solvent balance. However, NTM have multiple serovar-specific GPLs. This simply means that different species of NTM are going to have a slightly different GPL structure based on the glycosylation of various hexose-based molecules attached to a 6D talose residue. So now that we've talked about the analyte, we come back to bioreceptor. And before we can get into the details of that, one of the key things that we do in Dr. Bonham's lab is we study isoenergetic states. Simply, isoenergetic means having the same energy, and we use a predictive software called QuickFold that generates out two nucleotide sequences that have roughly the same thermodynamic value, meaning that if you just took a random solution, roughly 50% is going to be in one conformational state and 50% is going to be in a different conformational state. As you can see, the two states we picked here, on the left-hand side, we have a non-binding or an off conformation, meaning that within a sample that has our target MANLAM, it's not going to be bound. It's unable to be bound. On the right-hand side, however, if you do stick this aptamer sequence in a solution with target, it's going to bind, change conformation slightly, which is going to change this redox moiety and hopefully bring it closer to the gold-plated electrode. So originally, to detect MANLAM, we use this BM2 aptamer sequence. This was previously generated before our lab via selects, like we talked about, and it actually binds to the H37RV tuberculosis strain. So what we found was, while this whole aptamer sequence was used to bind to manlam in the past, the region with the greatest affinity was this N31 region right here. So we did a series of truncations. This is one of the truncations. And we found that if we truncated it, it actually had a greater dissociation constant, greater binding affinity to target manlam. Additionally, we inserted a methylene blue redox moiety tag right at its thymidine base, which is a key essential part of the signaling difference in electrochemical signals. Down here, you can see the actual aptamer sequence that we used. And after generating the sequence, we actually had to prove that it binds. So for proof of aptamer binding, we tried several different techniques, including electrophoretic mobility shift assays, spectrophotometry, milk curve analysis. However, none of these techniques really worked well at all. As you can see, here's the EMSA that we did, and it's very hard to differentiate what's going on here. So we turned our attention to different techniques, including fluorescence anisotropy. So fluorescence anisotropy basically measures the ratio of horizontally and vertically polarized emissions of light. So you have this cuvette with the aptamer sequence. We added additions of target manlam to it. And what happened was, as the manlam bound to the aptamer, the molecular weight increased, which decreased the rotational spin, which gave us a direct increase in anisotropy value. And I know it's a little bit complicated, but this graph, I think, does a good job of demonstrating that as we increased the concentration of target, the anisotropy value increased as well. So now that we proved this aptamer does bind, the next step is to go into voltammetric interrogation trials and to see if we can get a difference in electrical output upon addition of manlam. So what we found was that we did get a direct difference upon addition of manlam, and this is due to redox moiety. If you picture my hand as the aptamer sequence, and you have a redox moiety here, and this hand is a gold electrode, as the aptamer and the redox comes closer and closer to the electrode, that's going to increase the electrical signal, which is what we see here. However, unfortunately, as we did more and more trials, we found very inconsistent and inconclusive results. As you can see, sometimes the signal would actually be a signal off, meaning that the redox moiety is moving further away from the electrode. This could be due to surface probe density, meaning we might just have too much target or too much aptamer sequence that's saturated on the gold electrodes. We might try reducing the total aptamer concentration. Additionally, such things as temperature change could play a vital role in this, and it's something that we're actively pursuing right now. Additionally, while trying to increase the efficacy of the manlam trials, we're trying to purify glycopeptidolipids. Currently, we're trying to purify lipid extracts received from National Jewish Hospital in Denver, Colorado. Unfortunately, again, due to the amphipathic nature of GPL, it has made TLC plates very difficult to verify. Unfortunately, GPLs are too big for gas chromatography and slightly too complicated for carbon and hydrogen NMR analysis. However, after many, many trials, we think we're starting to improve technique, and we see a slight difference in, this is a MTB strain of lipid extract, and as you can see, a slight separation in possible cell wall constituents, and that's something that we're looking into and are really excited about once we're able to get back in the lab. The next steps of this project include in the short term, altering the electrode probe density to try to increase the efficacy of that man-lime aptamer. Additionally, we are verifying purification of GPL, and the next step is going to be to create an aptamer sequence using that selects technique. Long-term, kind of in summary, we want to use these biosensors for point-of-care diagnostics. This is really important because we can possibly cut down that 6 to 8 week long window to mere minutes. We're hoping that this can be a point-of-care diagnostic technique that could take probably under 10 minutes to accomplish, and that could really, really benefit some people who might not have access to medical laboratories. Additionally, we would like to pursue the use of these sensors for environmental detection, as that could be a very fascinating avenue to pursue. Real fast, I did want to touch on undergraduate research itself, as that's what this conference is about. It can be challenging at times. That's kind of an understatement. But see, I've run about 20 TLC plates with all of these different combinations in the past month or two here, and it's been insane. Almost none of them have worked. Additionally, my partner and I have ran about 50 gel electrophoretic mobility shift assay trials, and we've come to the conclusion that they just don't work, as you can see. All of this being said, research has been a life-changing experience for me, and one that I recommend for everyone. It has put me on a completely different career field and career track, and I'm very happy with what I'm doing and looking forward to getting back in the lab soon and continuing research again. Here are some of the references. And real fast, I'd just like to acknowledge Dr. Andrew Bonham for all he does for us in the lab, as well as for the past and present students of his lab, as can be seen up here. Again, thanks for the generous funding from MSU Denver and the support and supplies that we continually receive from Dr. Jennifer Honda at National Jewish Hospital in Denver.